Welcome to the series on food microbiology. In the last couple of lectures, we have seen how, depending upon the nature of the food we are talking about, the microbial flora change, what are the conditions which predominate when the microbial flora grow in them and proliferate in them, and what are the conditions which we modify to prevent their growth. In the second part of those lecture series, we saw, depending upon the nature of the food concerned, how the spoilage is attributed, how do we identify that there is a particular spoilage because of this particular organism in the particular food. Today, we are going to see the spoilage caused by microbes to animal products including milk, meat and seafood. Milk. As you all know, this is a part of your everyday life. And scientifically speaking, milk is the fluid excluding the colostrum which is secreted by mammals for the nourishment of their young. And colostrum is much more concentrated liquid containing up to 25% of total solids, and it is mainly protein. So, the principal components of milk are water, fat, protein and lactose. Human milk has lower protein but higher lactose content than cow's milk. Now, depending upon the category of mammal which we talk about, the protein and lactose levels will change. It depends upon the species we are talking about. And generally, the lipid content is comprised of C14, C16 and C18 fatty acids, mainly in the form of fat globules which are surrounded by a phospholipid rich layer, and this is called as the milk fat or the milk fat globule membrane. The basic milk protein is called as casein, and there are 5 main classes of caseins, and these caseins will aggregate together with calcium phosphate in milk to form colloidal suspension, colloidal particles. These particles we call as micelles. So, you have the basic 5 types of casein which are the alpha S1, the alpha S2, beta, gamma and kappa. These 5 main classes will form, along with calcium phosphate, a micelle. Then you have what you call as the whey proteins, the alpha lactalbumin, the beta lactalbumin, serum albumin and the immunoglobulins. So, this is actually the composition of milk. It is high amount of water, so its high water activity is present. The pH is actually very moderate, between 6.4 and 6.6, and it is a rich, nutrient rich source. So, all these factors make the rich excellent medium, a fantastic medium for the growth of microbes. And the common organisms which are isolated and associated with milk are micrococci, streptococci and the diphtheroid organism Corynebacterium bovis. If the animal is infested by mastitis, then the microbial count present in the milk, raw milk, would be very high. Okay? And this mastitis is an inflammatory disease of the mammary tissue, and this causes a major loss to the dairy industry. If you see the causative organisms of mastitis, you have the Staphylococcus aureus, E. coli, Streptococcus agalacti, Streptococcus dysgalacti, Pseudomonas aeruginosa and Corynebacterium pyogenes. All these can cause the same inflammatory type of disease manifestation in the cow or in the bovine which we are talking about, mastitis. And most of this or several of this are potential human pathogens also. And along with these, you have other human pathogens like Salmonella, Listeria, Mycobacterium also being reported in milk, which makes the milk a potential or an excellent nutrient source for the growth of microorganisms. And once the microorganisms grow, naturally the quality of the milk is going to get spoiled. What are the sources of microorganisms in the milk? One is the udder interior, 2 is the teat exterior and its immediate surroundings, and the milking and the milk handling equipment. So, along with this, you have to take into consideration the general conditions in which the animal is growing, how its diet is manifested, what is the hygiene level in the dairy farm we are talking about. Along with that, the immediate hygiene in that particular organism concerned. Okay? So, normally to prevent the microorganisms proliferation in milk, we give a heat treatment It can be a low temperature holding, which is called as LTH, which means 62.8 degrees centigrade for 30 minutes. A high temperature short time holding, which is called as HTST, 71.7 degrees for 15 seconds. Ultra high temperature, which is 135 degrees centigrade for 1 second. And the term sterilized milk means it has been kept for 20 to 40 minutes at a greater than 100 degrees centigrade. So all these treatments literally will lessen or reduce the amount of microorganisms which is present in the raw milk. Now, to test whether there has been a contamination, suppose you have pasteurized milk, you are keeping it, and then you want to know whether along with the pasteurized milk any other contaminant, like say for example plain water, it can be added. Water is also contains microorganisms, or suppose unpasteurized milk, raw milk has been added to this plain pasteurized milk. So then this will change the microbial flora present in the milk. So how do we identify this? For this, the common test which we use is called as the phosphatase test. This is a common test used to check the efficacy of pasteurization. Now, milk contains the enzyme alkaline phosphatase, and this alkaline phosphatase is extremely heat sensitive. So when it is pasteurized, the time and temperature which is maintained during pasteurization will inactivate alkaline phosphatase, which means this enzyme is inactive in pasteurized milk. So you add colored substrate, chromogenic substrate to milk. Okay, so this is your sample which you suspect which has been pasteurized, which you think it is sold. For example, say you think that it is sold as a pasteurized milk, but you suspect that there is a contamination of this by raw milk or maybe some other contaminant. So to this supposed sample, you are going to add a chromogenic substrate. Remember, if the milk has been pasteurized and is uncontaminated, the alkaline phosphatase present in the milk would be inactivated. So when you add the chromogenic substrate, it will be colorless because the enzyme is not active. Whereas if there is a contaminant, active phosphatase is present, then this chromogenic substrate will hydrolyze to produce a color forming sign. So it will produce a color. The presence of the color indicates that there is a contamination. Raw milk has been mixed or unpasteurized milk has been mixed with your pasteurized sample. The same principle is used in the case of egg, bulk liquid egg. We check the alpha, which is called as the alpha amylase test there. We will see that when we are talking about contamination of egg products. In milk, which is subject to very low levels of post-pasteurization contamination, the usual spoilage organism which grows as bacillus, which is thermoduric, because it can withstand the pasteurization temperature. The growth of this bacillus usually is associated with flavor defects, but in most of the cases this is called as the bitty cream phenomenon because of the lecithinase activity of the Bacillus cereus. Now, what happens is that Bacillus cereus produces lecithinase, and milk contains phospholipids in the milk fat globule membrane. So this lecithinase enzyme will hydrolyze the phospholipids associated with the milk fat globule membrane to produce proteinaceous fat particles. These proteinaceous fat particles will float on the surface of hot drinks. It will stick onto the surface of the vessels or the crockery or the glasses. So you will have patches of fat particles sticking onto or floating on the surface of your hot beverage, your tea, coffee, etc., or it will be attached to the surface of your glass, the crockery, the vessels. This indicates, this fat globule attached there indicates that there is proliferation of the Bacillus cereus in the milk sample. This bitty cream is actually associated with milk that has been subjected to temperature abuse, meaning after pasteurization, the holding temperature, there has been a change in the temperature where it has been stored during transportation, or maybe the stability of the temperature during pasteurization was not maintained, as a result of which you had the proliferation of the microorganism. The psychotrophic Bacillus species are becoming increasingly associated with spoilage of refrigerated milk. So this, even though not much disflavor is felt, you will see the flat globules fatting, which is also associated with microbial growth in milk concerned. Meat and meat products. Now, This again is a major animal food where you have a large scope of contamination by microbial growth. What we call as meat or edible animal flesh principally comprises of the muscular tissues, but also organs like as heart, liver and kidneys. So step by step we have to see at which particular time we are going to have maximum amount of microbial contamination happening to the meat product. So this we have to start stages of animal slaughter. So when the animal gets killed, number one, the circulation will stop. So when the circulation stops, the ability of the animal to synthesize ATP is lost. So ATP is the energy producing molecule in all animals, in all cells concerned, it is required. So you no longer have ATP production in the organism because it has been killed. This lack of ATP will cause actin and myosin to combine to form actomyosin, which leads to stiffening of muscles. If you remember the basic muscular contraction relaxation, which involves actin and myosin, every time you want to have a muscular contraction relaxation, ATP gets hydrolyzed. And that ATP is normally provided by the cell concerned. This is normal. So when ATP is no longer synthesized, the actin and myosin will join together to form the actomyosin, but there is that easy contraction relaxation will not happen because there is no longer a source of ATP there. So this will lead to stiffening of the muscles concerned. No respiration, so the oxygen supply will fall. This will result in the reduction of the oxidation reduction potential. Now the next step what happens is the supply of vitamins and the antioxidants will cease, will stop. This will result in a slow development of rancidity. So the muscles have become stiffened. There is no oxygen supply, so reduction in the oxidation potential, reduction potential. 3. The antioxidants are no longer present. The vitamin supply is no longer present. The nervous and the hormonal regulations. So even if a cell is supposedly under resting condition, not moving or immobile, you have the lot of background activities like your ATP production, the hormonal regulation, circulatory system responding to the stimuli, external stimuli, all that happens even in a resting cell. But here, because the animal has been killed, all that will stop. So the nervous and the hormonal regulations will cease, thereby causing the temperature of the animal to fall. So the automatic temperature regulation to the normal body temperature will also stop. So temperature will start falling. And as a result of this fall in the temperature, fat will start to solidify. Complete stop of respiration, which will stop ATP synthesis. Now, because of all these conditions happening, you will have glycolysis starts. And the stored glycogen will get converted into lactic acid. Production of acid will reduce the pH. So it will depress the pH to the normal 7.4 to ultimately it will become 5.6. So you will have glycogen getting converted into lactic acid, reducing the pH from 7.4 to 5.6. Now at this level of pH, protein denaturation will start. So pH depression in turn will stimulate or initiate protein denaturation. This will liberate and activate cathepsins, and this in turn leads to what we call as the onset of rigor mortis. Protein denaturation is also accompanied by the exchange of divalent and monovalent cations on the muscle proteins. So all these changes happen when the animal gets killed. The reticular endothelial system also ceases to scavenge. So you don't have the immune system of the animal now active, trying to reduce the amount of microbial or check the microbial growth. So when the checkpoint immune system regulation of the microbial growth is stopped, automatically the microbes will start proliferating in the system because there is no control and meat is still a nutrient rich source for the microbial growth concerned. So the microorganisms will grow unchecked. Various metabolites will start accumulating, which also aid in protein denaturation. So step by step you are having a conducive change in the slaughtered animal, which in turn will make microbial growth much more rapid. In general, the biota is reflective of the slaughtering and the processing environments, with gram-negative bacteria being predominant. Now, the amount of microbes which grow depending upon when, number one, the health condition of the animal, as we said, two, the hygiene, the sanitization and hygienic conditions where the animal is slaughtered and then the meat is processed, and normally it is the gram-negative organisms which predominate in such scenario. You do have some gram-positives which grow up. The enterococci are the ones which are more often found along with lactobacilli. Because of the ubiquity in the meat processing environments, you see a large amount of moles, including penicillium, mucor, and cladosporium. To delay the microbial spoilage, fresh meat is normally stored at refrigerated temperature less than or equal to 5 degrees centigrade. Unless this facility is not available, it's preferable to store at refrigerated temperature. So, in this temperature, what you are going to see is the growth of psychotrophic bacteria. So, raw meat spoilage organisms are mostly psychotrophic, which can grow in the range in less than or equal to 5 degrees centigrade. And in retail cuts, because you keep them for a shorter generation, you are going to see the growth of Pseudomonas species. And these Pseudomonas will first utilize glucose as a carbon source, then it will start using the amino acids, and the metabolism of the amino acids is then accompanied by the production of methyl sulfides, esters and acids. So, step by step you are going to have the spoilage of the meat concern. This is normal. Now, when we let's see what is going to happen to the ready-to-eat meat products. So, as you are all aware, the ready-to-eat meat products would include high heat processed and low heat processed, uncured and cured meat products. So, depending upon to which category your ready-to-eat meat products belong to, the type of spoilage which is manifested is going to be different. If it is cured and high heat processed measles, normally we can again give them heat treatment to make them commercially sterile. So, when they are again given heat treatment to make them sterilized, what would be remaining there would be the spores, the thermophilic spores. Remember, spores will not get spoiled easily. So, unless these germinate, conditions become conducive for the germination of the spores, you are not going to see live vegetative bacteria in these ready-to-eat products. So, comparatively they are safer. The growth of the spores will happen when this has been taken out from the storage temperature and it is kept out for thawing and without utilizing it completely. If you keep it back because of the temperature abuse, you are going to see the growth of the thermophilic spores there. Unpackaged cooked products that do not have carbohydrates can become putrid because of the growth of and protein degradation of the proteolytic gram-negative bacteria. So, these meat products do definitely contain proteins, and you have the proteolytic bacteria which are capable of breaking them down. So, this will give rise to a putrid odor. And if the products are stored for a long time, yeast and mold can also grow, causing off flavor, discoloration and sliminess. So, this is going to be the condition when it is stored for a longer time, but for a shorter time this does not manifest. And because of the growth of hydrogen peroxide producing lactic acid bacteria, the meat will start showing green to grey discolorations, which indicates that spoilage has started. It is better not to eat the product. We are going to now see egg and egg products. Egg, normally raw egg, because of this its outer shell made up of calcium carbonate is comparatively safe, but you do have pores which are present on the egg membranes, and these pores in the eggshell and the egg membrane, inner membrane, they do not prevent the entrance of bacteria, and also if the spores have germinated on the outer side, through the small pores the hyphae of the mold can grow inside, and as the pore size increases, which happens during storage, if you are going to keep it for a longer couple of times instead of utilizing it immediately, the pore size will increase, then you are going to see more bacterial growth, and the hyphae will in turn go and start spreading inside the eggshell. The presence of moisture also enhances the entrance of mobile bacteria inside. The albumin and the yolk have 0.5 to 1% of the carbohydrate. They are very high in proteins, but they are low in non-protein nitrogen. But still, even though it's low in non-protein nitrogen, because they are very high in proteins and rich in carbohydrate, you can still have bacterial growth there. And during storage, the pH can shift to the alkaline side from 9 to 10. In addition, egg has lysozyme, the enzyme. This lysozyme causes normally the lysis of the bacterial cell wall and the mucopeptide, the conalbumin which chelates the iron, antivitamin proteins, for example avidin binds to the riboflavin, and protease inhibitors in the eggs normally will prevent microbial growth. So, raw egg naturally has components which can prevent microbial growth. For example, the lysozyme, which can, suppose you have a microbial contamination, the lysozyme can break down the bacterial cell wall. Now, the conalbumin can chelate iron. Remember, most of the microbes have to get an iron from outside. So, if iron is in a chelated form, it will not be available for the organism to uptake. Then, avidin. It has avidin which will inhibit riboflavin, so the B complex proteins will no longer be available for the organism to uptake. Then, it also has protease inhibitors which prevent the microbial growth. So, normally egg has a sort of protection, but when the microbial contamination becomes more, then as it gets stored for a longer time, these protective measures will get neutralized. The most predominant spoilage of shell eggs is caused by gram-negative motile rods from several genera that include Pseudomonas, Proteus, and Alkaligenins. And the different types of rot which we see, we designate them as rot. For example, the green rot causing the greening of the albumin because of the growth of Pseudomonas fluorescens. The black rot producing muddy discoloration of the yolk. So, in the first case, Pseudomonas changes the color of the albumin to green. In the second case, it is manifesting the protein rich yolk mainly because of hydrogen sulfide production by Proteus vulgaris. Then you have the red rot by Serratia mercescens caused by the production of red pigment. So, this picture shows the black rot, for example. You have the green growth because of the presence of Pseudomonas here. So, these are the normal damages, microbial spoilage, which is seen in raw eggs. And the liquid eggs, which contain the whole yolk or the white. When you break open the shell and you are compiling the components combined together or in a separate form, generally we pasteurize them and then freeze them, or either pasteurize or immediately freeze, both are done. So, this procedure will normally inhibit microbial growth. So, you separate out the egg compiled, the albumin and the yolk combines together, albumin separate or the yolk separate, you separate them together, pasteurize and freeze, or either pasteurize or freeze. So, this is the way it is normally stored, the liquid part of the egg. Now, if the liquid products are held at room temperature following breaking prior to pasteurization, that is, you have broken the egg and separated out, but it is still kept outside at the room temperature before pasteurization. So, that exposure at room temperature can make the spoilage bacteria grow in the liquid, causing off flavor, that is, proteins will get denatured, proteolytic activity, sourness or fishy flavor due to the formation of trimethylamine. So, break it open and immediately pasteurize or freeze. You cannot keep it exposed in room temperature, otherwise the product will get spoiled. Pasteurized eggs at refrigerated temperature will also have a limited shelf life unless you add additional preservatives, which means that eggs, which are a very rich source of nutrients, are an equally rich source of nutrient for microbial growth. So, unless proper precautions are taken, it cannot be stored for a longer time, whether pasteurized, whether frozen, or both together, combined together. Fish, crustaceans and mollusks. So, seafood generally form a different category, separate category, where you have to take different sorts of precautions, either fresh, that is fresh water, or whether it is salt water, marine environment. In both the cases, the nature of the nutrition component of these products are totally different from what you see in a land-borne animal meat. Fish, whether it is harvested either fresh water or salt water, normally here we have the autolytic enzyme actions. They have a large amount of unsaturated fatty acids which are present in the fish. So, these unsaturated fatty acids will start getting denatured the moment you take them from the mammalian environment and they become dead. And this in turn will also cause the proliferation of microbial growth. So, autolytic enzymatic actions, the oxidation of the unsaturated fatty acids, and in turn microbial growth. These are the three major aspects of damage which happens to fish, crustaceans and mollusks. Now let's see step by step the type of spoilage which manifests. Protein hydrolysis by autolytic enzymes, the proteinases, is predominant unless the fish is gutted immediately. The moment it is taken out following a catch, it has to be gutted, otherwise autolytic enzymes will become activated. Second is the oxidation of the unsaturated fatty acids, and this is very common. This happens irrespective of whatever be the condition you are storing it in. And three, the microbial spoilage. It is determined by the microbial types, their level, the fish environment, the type of the fish, the method used for harvest and subsequent handling. So, what is the type of fish you are talking about? Its microbiota will change. So, depending upon the changed microbiota, the spoilage cause is going to be different. Then, normally in the environment, whether freshwater or marine, what is the level of microbial contamination which is present? You have a normal level which is present in water. If the microbial load is higher than normal in the environment, naturally the organism will have higher than amount of the normal microbiota, and this higher than amount of normal microbiota in turn will cause rapid spoilage of the fish concerned. And method of harvest and the post harvest storage conditions will also affect the spoilage. Fish tissues have high levels of NPN compounds, which we call free amino acids, trimethyl amino oxide and creatinine, in addition to peptides, proteins, but almost no carbohydrates. The pH is generally 6.0, and because of the relatively shorter generation time, here it is the Pseudomonas species which predominate, the psychotrophic, because you refrigerate our harvest, but the refrigeration temperature, which is less than or equal to 5 degrees, will allow the psychotrophic Pseudomonas to grow. So, normal aerobic storage, whether it's refrigerated or slightly higher temperature, the spoilage causing organism is going to be Pseudomonas. And if it is stored under vacuum or in carbon dioxide, the lactic acid bacteria including Enterococcus can be predominant. So, depending upon the conditions of storage, you are going to see changes in the type of microorganisms which predominate. In fish stored by vacuum or by carbon dioxide packing, the growth of aerobic spoilage bacteria is prevented, but you also have anaerobic and facultative bacteria which can grow, including lactic acid bacteria. And under refrigerated condition, they will have a longer shelf life due to the slower growth of the spoilage bacteria. And canned fish, normally we give them heat treatment to produce commercially sterile process, and here the spore formers are the only case where which can cause spoilage. In the current session, we saw what are the predominant organisms which can cause spoilage to animal meat products, the egg products as well as the fish. In all these cases, depending upon the predominant microflora which are present in the animal concerned at the time of slaughter or at the time of catch, the type of spoilage will vary, and depending upon the immediate storage process which happens, you can extend the shelf life by reducing the amount of microbiota which grow there. Thank you.